Alright, hello everyone. Today we're going to be talking about habitats, microhabitats, and species distribution. So an ecosystem is not uniform. It contains a range of habitats and microhabitats, and this directly affects how species are distributed and how reliably they can be sampled. So what's the difference between habitats and microhabitats? A habitat is the environment in which a species normally lives. So it's defined by its physical, chemical, and biological characteristics. But within a habitat, microhabitats are smaller, localized areas with distinct conditions, such as the underside of a rock, a patch of shade, or the base of a tree trunk. So just think habitat, broader environment, microhabitat, localized area. Species are not distributed evenly across habitats. Their presence and abundance in a given location is determined by how well the conditions of that habitat or microhabitat meet their biotic and abiotic requirements. So we've talked a little bit already about the patterns of species distribution. So species distribution across an area typically follows one of three patterns. So we have uniform distribution, and that's where individuals are evenly spaced. And this occurs when competition for resources is high and individuals actively maintain distance from one another. And then you have random distribution. And just as you might guess, individuals are scattered without a predictable pattern. Now, this happens when resources are evenly available and individuals do not interact strongly. And then you have clumped distribution. Individuals are clustered in groups. This is the most common pattern in nature, and it occurs when resources are patchy, when species aggregate for reproduction or protection, or when microhabitats concentrate suitable conditions in specific locations. Now, the distribution pattern of a species directly affects which sampling method is most valid and reliable. So if we're looking at randomly distributed species, we're going to use random sampling. Okay, quadrats placed at random coordinates will provide a representative sample. So random distribution, we use random sampling. For uniformly distributed species, systematic sampling. Okay, so that means placing quadrats or transects at regular intervals captures the distribution accurately. Now for clumped distributions, neither random or systematic sampling may adequately capture the full range of variation. Stratified sampling, which divides the area into distinct zones based on the habitat type and sampling each proportionally, is most appropriate as it accounts for the patchy nature of the distribution. So we're going to use stratified sampling for clumped distribution. So failing to match the sampling method to the distribution pattern introduces bias. Some areas or habitat types may be overrepresented or underrepresented, reducing the validity and reliability of the results. So to summarize, ecosystems contain a range of habitats and microhabitats that create variation in environmental conditions, influencing where species are found and in what abundance. Species distribution may be uniform, random, or clumped, with clumped being most common due to patchy resources and microhabitat variation. The sampling method must be matched to the distribution pattern. Random sampling for random distributions, systematic for uniform, and stratified for clumped to ensure valid and reliable data. Follow for daily QCAA content.